Okay, so we're going to start this discussion at the shared ancestor for all of the chordates. And going backwards from this, you would basically go back to the deuterostome common ancestor that we share with the starfishes and the other echinoderms, the deuterostome common ancestor. So from the chordate common ancestor, from the chordate Merca, we're going to be taking two roads. We've got a most basal of all chordates, and this is going to go to the modern group known as the cephalochordata. Most basal branch, cephalochordata. And the other branch, the other main group, the other larger group of chordates is going to go down the main road toward the vertebrate lineage, but we're not going to be quite there yet. But before we get there, we need to talk about cephalochordates a bit. Cephalochordates are really kind of neat. We've got some modern representatives. Sometimes cephalochordates are called lancelets. Sometimes they're called amphioxus. Amphioxus is the old scientific name for them. Sometimes we call them branchiostoma because that's the modern scientific name for them. But basically, they're just cephalochordates. And when you look at a cephalochordate, it's a really pretty simple thing. I'll draw you a picture of one. It's got kind of like a loby mouth, a mouth on one side that will be sucking up food. It's got a long knife-shaped body, right? Kind of fish-like. It's got a muscular tail. You got little, units of segmented muscle that go down the tail, but it's able to wag. There's a little fin-like structure along the back. It wags this and creates movement, allows the little thing to swim forward, swim backward. It actually can go in both directions, right? It doesn't have any eyes. It doesn't really even have a brain so much. It does have a little bit of a head. You can tell this end is on the head end, and it's got a central nervous system that is just barely there. I mean, it's a dorsal structure. It's got a hollow space on the inside. Yeah, so in other words, it's dorsal and hollow. We can call it a nerve cord. And that's what we're seeing as kind of like the central nervous system for our amphioxus. We've got a muscular post-anal tail, which is what I'm telling you, which I told you about before. Let's say it's post-anal because it actually extends behind the vent. And if we were to draw the digestive system in to this animal, you would have like intestines over here. I'm drawing this backwards. So the intestines would connect to a stomach. So a stomach's over here. And so maybe this is the animal. And then the stomach would connect through an esophagus to the pharynx of the animal, the throat of the animal, which would then connect to the mouth. So you've got this long area of a pharynx. Here's the anus. And you got the pharynx. And the other thing about this is that we've got on the outside part of the structure, remember that the pharynx is a tube that runs down. This is called the front part of the gastrointestinal system. You've got the mouth, pharynx, esophagus, stomach, intestines, anus. On the outside of the animal, we've got slits, either slits in the case of amphioxus or pouches like in the case of a lot of other animals, slits that connect the outside world to the pharynx. And what happens, this is really significant because the animal is taking in food together with the water that it sucks up from its mouth. It draws in lots of water through its mouth. The water gets pushed out through these pharyngeal slits, and the food gets trapped inside the esophagus, and the food then goes down to the stomach where it gets digested. So we've got these pharyngeal slits. And right there, I've given you 3 out of 4 of the chordate synapomorphies, the characteristics that are presumably shared by all chordates from their most recent common ancestor, probably sometime back in the later part of the Precambrian. Okay, that's 3 out of 4. The one that actually gives the chordata its name, however, is this thing. It's kind of a semi-rigid insert. It's like a rod that goes down the length of the body of the animal, kind of like where your vertebral column is, or visualize the vertebral column, the vertebrae of a fish. And that's approximately the location of where the notochord would be. And what it does is it keeps the animal relatively rigid, and so it's able to have kind of like a torpedo-like shape. It doesn't flop around flimsily. That's kind of the characteristic of these chordate worms, like the amphioxus. It's not as floppy as like a red worm. It's more like a stiff-bodied worm that wiggles this area over here, just under its tail, to move around, and that's the notochord. And so if you look at some of the earliest fossils that we have of chordates, like Unanozoon, from the earliest Precambrian in China, they look more or less exactly like these amphioxus that we have today. In other words, there's been very little morphological evolution in the lineage that's given rise to amphioxus. Now, cephalochordates like amphioxus slash lancelets, oops, this should say lancelets, okay, not lancets. Lancet is actually a device used by a surgeon. Okay, so where was I? Oh, yes. So cephalochordates are certainly chordates. They're part of our phylum. They're one of two groups of clear invertebrates that are part of our phylum. It's not that chordates and vertebrates are the same thing. Chordates include both vertebrates as well as invertebrates. Cephalochordates are one kind of invertebrate. The other one is the second branch off from the main road here. So we're going to put another branch off here, and it's going to go to another group of relatively basal chordates, not quite as basal as the cephalochordata, but this is one that we'll be calling the urochordata. Urochordates include things like tunicates, are more or less the same thing as ascidians, and you've got larvaceans. But the main group of urochordates we talk about are the tunicates, and what they look like is kind of weird. They've got this particular anatomy. They're really easy to recognize because they've got two holes and they're sessile. So if here's a rock face, sometimes you'll find these things that are attached to the rock, and they're going to have like two siphons. There'll be a siphon over here and a siphon over here. And that's the urochordate. That's all it is. It kind of looks kind of like a sponge, but it's not a sponge at all. It's actually pretty highly derived bilaterian, and it's actually kind of a close relative to us. Now, the basic anatomy of a tunicate is kind of important for us to get into. This siphon, usually the longer siphon, is called the buccal siphon. And so the water is taken in through the buccal siphon and it goes down, 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 down into a pharynx. And so there's a pharynx, which is kind of like, it might kind of make sense. It's connected to a mouth, and the pharynx is connected to an esophagus, which is then connected to a stomach. Okay, and then the stomach is connected to intestines, and intestines are going to be coming out in this direction. Okay, okay. Now the other part of the urochord, I'll draw it in a little bit of a different color, is going to be the atrium. And so this site over here is going to be called the atrial siphon. And the atrial siphon will then connect to a pretty large space that overlaps with the pharynx. Okay? And so the atrial siphon is the space that connects to the outside world. And on the pharynx, we've got these little slits which create a pharyngeal basket. It's going to be the pharyngeal basket. And so the circulation of water is like this. Water is going to be coming in through the buccal siphon. It's going to be coming into the pharynx, and the water is going to be squished out. Basically, the water is going to be escaping through the pharyngeal slits from the pharyngeal basket into the atrium. And that's going to be creating positive pressure in the atrium. Then the water is going to be moving out through the atrial siphon. In the process, all the food that is trapped inside the pharyngeal basket, think of it like a colander, like the colander that you use to strain pasta. All that food is going to be passing into the digestive tract. So the food is going to be ending up here. The animal is able to eat, and the anus is basically located strategically close to the atrial siphon's opening to the outside world. In effect, this thing is very similar to a living aquarium filter. It basically draws water in, cleans out all the particulate matter, and then it pushes that water out. The anatomy of the adult urochordate is not especially that important to you. What you want to think about here is that this animal is actually kind of closely related to you. It's a chordate. How do we know it's a chordate? Well, it actually has some characteristics that let us know that. Not so much when they're in the adult state, okay? In this adult urochordate, you don't see any chordate characteristics. You don't see a notochord, you don't see a muscular postanal tail. You do see pharyngeal slits. The slits in the basket are pharyngeal slits, but you have to back up a little ways in order to actually see how this animal corresponds to the cephalochordate body plan. Okay, so if you go to a larval stage of a urochordate, it looks kind of like what we have in the case of a cephalochordate. Right? It's going to have a head, it's going to have a little mouth, it's going to have a long tail, and the tail is going to contain a urochord, which is actually a notochord. Right? There's going to be pharyngeal slits. There's going to be an anus right there. So in other words, this tail is going to be muscular and postanal. And what happens is that this weird little animal, is going to settle down on the bottom. It's going to transform. It's going to land right here on its head, and it's going to transform into the adult urochord that we see over there. In other words, you look at the adult, you don't see anything that looks chordate-like. But if you look at the larva, it's definitely a chordate. And so, yeah, I mean, sometimes you've got to look a little bit past the adult morphology. Now, going back to this main road, I mean, apart from the urochordates, the other major group that falls out from that main road are the vertebrates, the vertebrata. And there are really two characteristics that I want to highlight when it comes to understanding the vertebrates. First of all, I want you to realize these earliest vertebrates had no jaw. These were jawless. What they did have were, well, obviously vertebrae. And the other thing that they had was a new kind of head. I'm going to call it the new head because that's the term that's actually used in zoology. So let's start with the vertebrae. The vertebral column consists of separate units, and each unit is called a vertebra. Okay, and a vertebra consists usually of some type of cylindrical barrel. There's a centrum, that's the main part of the vertebra. Then you've got a little space on top of that, which is going to accommodate the spinal cord. And sometimes, for example, in the case of a fish, you'll have like a neural spine on top and a hemal spine that'll come off from the bottom. Sometimes there'll be spaces in here that will accommodate blood vessels. But the basic unit of the vertebra is going to be some kind of support structure. That's the centrum, right? And that's going to be lining up with other vertebrae, right? Okay, you'll have another vertebra over here with its own little space above it, which accommodates the spinal cord, its own neural spine, and a hemal spine. And the spinal cord is going to pass right through here, right? The spinal cord is going to be going from one spinal canal to the next spinal canal. And one feature of the vertebra is that it provides a little canal, a little protection, a little space in which the spinal cord can be protected from pressures from the outside world. Okay, and so that's one feature. That's the vertebra, and all vertebrates have this. It's different from it's different up here. When you look at the cephalochordate, you see the dorsal hollow nerve cord is there. It's sitting more or less on top of the notochord, but there's nothing wrapping around it. Okay. The other thing is that the vertebra is pretty much taking the place of the notochord. Instead of having a notochord here, you're going to have the centrum, the vertebra, the spinal cord is going to be going in here. We're not going to be needing the notochord anymore. In fact, in vertebrates, what we find is that the notochord gets repurposed. The tissues of the embryonic notochord often get repurposed as the stuff that occurs between the vertebral centra. We call them intervertebral discs. And part of that disc is deriving from the cells that were once part of the embryonic notochord. Okay, so that's one characteristic of the vertebrae. The other characteristic of the vertebrates is this thing we call the new head. And in order to understand this, we've got to understand a population of cells called the neural crest cells. And no, the neural crest cells are not going to become the new head. They're going to be directing a whole bunch of developmental events, part of which is the development of a head in a brand new way that's never been done before in any other form of animal. Okay, so let's take a little bit of time to talk about, well, what these neural crest cells are. So thinking about neural crest cells, what we need to do is we need to back up a ways. We need to back up to think about the development and the formation of that dorsal hollow nerve cord. And I want you to think about a gastrula. And so in the case of a vertebrate embryo, we're going to be seeing a gastrula. It might look like something like this. And here's the archenteron on the inside. And here's the mouth that's going to be forming over here. The mouth is going to be on this side. This is going to be the anus. On this side, mouth, anus. And ultimately, we're going to be forming a dorsal hollow nerve cord in this area right here. Right? That's going to be the dorsal side. Now, the source of that dorsal hollow nerve cord is going to be coming from a stripe, kind of like a skunk stripe of ectodermal cells right on the top of the embryo. These cells are going to be sinking downward, and they're going to be forming a neural tube. So if we turn this guy around, turn this guy 90 degrees so he's facing us, sort of visualize this stripe, this skunk stripe of cells on top of the embryo. The rest of the embryo is like this, and the and these cells at the very top are going to be dipping downward, going to be forming a little neural groove, and ultimately they're going to be forming a neural tube. So there's a neural groove, and when that finally dips down, it's going to be forming a little neural tube. And that's going to be the dorsal hollow nerve cord, right? It's going to be forming like right in this area here, right? So that's where the dorsal hollow nerve cord is going to be forming. All right, and so what I want you to focus on here are a population of cells. I'm going to draw them in green. Okay, and so these cells, they kind of start off right there at the juncture, right there, kind of like between the neural ectoderm and the regular old ectoderm. And as these cells at the boundary between the two areas are taking part in this process of neurulation, the formation of the neural tube, they end up kind of in limbo. These cells are kind of halfway, or they're kind of stuck between the neural tube and the dorsal epidermis. Okay, these are the neural crest cells, NCC. And what happens is that they start migrating all over the animal. Okay, let me draw this again. So here's our little embryonic fish. Here's a dorsal hollow nerve cord. And these neural crest cells in here are all going to be starting to migrate. It's fascinating to watch. They migrate all over the place. A lot of them are going to be migrating towards the front of the animal, towards the front, and some of these are going to be forming a cranium, a little brain case, a little space to cover the brain, right? So the formation of a skull is going to be happening as a result of the direction that's given by the neural crest cells. The neural crest cells that migrate forward are going to be giving rise to a cranium, are going to actually direct the formation of a cranium. Others of these neural crest cells are going to be migrating down, and they're going to cause a formation of a lower jaw. Right? Others of these cells are going to be migrating out here, forming the sensory structures. They're going to interact with some ectodermal placodes. Basically, these neural crest cells are incredibly active. They migrate all over the embryo, and they do fascinating things that are really kind of unique, entirely unique to the vertebrates. Right? But most importantly is the head. Basically, these neural crest cells are going to be defining a zone in front of the animal. It's going to be taking a particular pattern of development that's different from anything that we see in the rest of the animal kingdom. And um, and people have been recognizing this for a long time. I think this is sometimes one of the keys to understanding how vertebrates are distinct from the rest of the animal kingdom. Now we're going to continue the tree. I've got to move it over to the left-hand side because I run out of space really quickly here. And so from the vertebrata, I'm going to draw one line upwards, which is going to be the line which corresponds to the animals that retain that characteristic of being jawless. Call them jawless fishes. And I should actually draw several different jawless fishes. There are probably several different lineages that are distinct from each other. One of these lineages of jawless fishes is still alive today. This is one that gives rise to modern day hagfishes as well as the lampreys. Okay. Your book might refer to these guys as the Myxini, Sometimes you could refer to the lampreys by a really long name, the Petromyzontiformes. I'm not even going to write that down. But you've got a couple of modern fish that are kind of eel-like. They're really kind of weird. Lampreys are parasites. They attach to fishes and they suck the blood out of them. Hagfishes are little worm-like things that creep around on the bottom of the ocean. They tear apart carcasses that of larger animals that die and settle out of the bottom. So you got a couple of weird things that live today. If you go back to the Cambrian, there are other kinds of jawless fishes, really large things. I mean, the Anapsida that had really long tails. Characteristic of these guys is often that they didn't really have too many fins. Um, Sometimes they had like a single fin, but they're characterized by having these really heavy plates of armor, right? They're heavily armored fishes, these jawless fishes of the early times, and this long piece in the back would basically be like a gigantic spike. It'd be covered by armored plates, and the armor that we have here, these armors are also presumably directed by these new kinds of cells, the neural crest cells, the dermal plates or things that are akin to the dermal plates today, like the scales of fishes. Their development is directed by the neural crest cells. Okay, so these jawless fishes were actually very abundant and very diverse in the early days of vertebrates, but they eventually got outcompeted by the other group of vertebrates, which is again, it's the main branch of vertebrates, and this is the Gnathostomata, starting with a silent G. Gnathostome refers to the fact they've got a jaw. Stoma, you probably remember that means mouth. And so these are the jaw-mouthed vertebrates. Now you got to realize that having a jaw that articulates, that can accommodate teeth, represents a huge innovation in the history of vertebrates. I mean, what it does is it allows animals to immobilize prey. If you think about these early jawless fishes, they couldn't really do much. The most they could do would probably be something like filter feed. And so a filter feeding jawless fish wouldn't need to have too much brain power. You don't need to be you don't need to be an Einstein in order to just kind of cruise along the bottom and hoover up whatever is in your way. You don't have to worry about predators too much because you got this big armored plate. That's not the life of a brilliant fish. Once you become a predator, you've got to outsmart your prey items. And one of the things you have to have here is a jaw. Okay? And so what I want to do is I want you to think about, well, how you could do a transformation from a jawless fish to having a jaw. And this is actually going to involve some of the neural crest cells. And it's also going to require us to think about something we talked about a while ago in one of the earlier units. This is the idea of homeosis. The idea that sometimes we have repeated body parts. And if we have extra copies of a repeated body part, they can sometimes be repurposed. And so visualize for an instant a fish whose front end kind of looks like this. Right? Here's the front end of a fish, and this is a jawless fish. Right? There's a mouth in the front, but no jaw. Okay. You also have pretty large pharyngeal gill slits. These slits are there. They are allowing water to pass out. The food that's trapped inside of the pharynx is going to be swallowed. You know that story. Now, part of this picture is going to require there to be these rods, these skeletal rods, cartilaginous rods that support these flaps of tissue between the pharyngeal slits, right? We see these in our laboratory. When we go to dissect fishes, we'll actually see that there's an upper and lower limb to the gill arches. These ultimately become the gill arches of fishes. Okay, but we've got several copies of them, and these skeletal rods are kind of like the starting point for the formation of a jaw. You might even be able to see this. All we have to do is we've got to get this piece of cartilage, this one right here, to extend a little bigger. We're going to make it migrate forward, and that's going to become the lower jaw. This piece here can actually become articulated. It's going to become the upper jaw. And so if you fast forward this a few millions of years, what you might be able to envision is that there's going to be an upper jaw, which is going to be consisting of that top piece. Okay? You'll have a lower jaw. It's going to be consisting of the bottom part. We actually call that Meckel's cartilage. And that's going to be the parts of the fish that allow it to articulate and immobilize prey, and maybe their teeth. Now, all this is required is that we've repurposed those skeletal elements in that first gill arch. We still have all these others to carry out the same function that we had before. So in other words, we're still going to have gills, gill slits, right, pharyngeal gill slits, and they're still going to be supported by skeletal rods. We're just going to have one fewer than we had before. The first set of skeletal rods is going to be repurposed to form the jaw, right? And so what happens if you have a jaw, you become predatory, you start becoming smarter, you start depending on having really good vision. You need to maneuver around better, so having paired fins that allowed you to stop and start and steer very carefully allow you to capture prey items. The whole ecology of the animals changes once we become predatory, right? And so this is kind of like a major landmark in the evolution of vertebrates. Now, looking more carefully at our tree, again, looking at the gnathostomes, we're going to be seeing three major clades of gnathostomes, one of which is entirely extinct. We've got the placodermi, which look more or less like this guy here. Okay? Placoderms were around during the Paleozoic, and they went extinct during the Great Dying at the end of the Paleozoic, right there at the Permian-Triassic boundary. There's another group of gnathostomes that's still around today, a very highly successful group. This is the chondrichthyes, also known as the cartilage chondroichthyes fish, the cartilage fish. By this I mean, yeah, sharks, rays, skates, chimeras. It's a highly successful clade of marine fishes, but they're not the group that represents the main branch off the Gnathostomata. The main branch here is going to be the osteichthyes, also known as the bone fish, bony osteichthyes, bony fish. Sometimes people will refer to this group, this larger clade, as the teleostome. Uh, teleostomy. And this refers to the fact that the osto, the bone, is in its ultimate form, ultimate bone. Or sometimes we might even call this true bone, in contrast to the kind of bone that we have in the other kinds of gnathostomes. Chondrichthyans, they're called chondrichthyes because they don't really have any bony skeleton. They've got cartilage. Sometimes the cartilage is pretty well calcified, but it's still basically just calcified cartilage. In these early jawless fishes, we've got these bony plates, these really heavily armored bony plates. And you might call that bone, but it's not true bone, it's dermal plating. It's basically a bone-like structure that forms on a two-dimensional surface. In the case of true bone, what we're really talking about is something that anatomists will sometimes refer to as replacement bone or endochondral bone. Sorry for the plethora of different terms. And what we're really talking about here is like a three-dimensional structure, a three-dimensional bone that we find in the long bones of vertebrates. So if you were to look inside of your humerus, your long bone, which is in your upper arm, there'd be a solid, really bony, hard, three-dimensional structure to most of the bone. You'd have the ends, the part that actually articulates with the shoulder on one side, a little ball and socket joint, and you've got the other side. It's going to articulate with your lower arm, the ulna, and the radius. But the actual structure of this bone, if you look at it really, if you look at it at a microscopic level, what you find is this three-dimensional matrix in which you've got these osteocytes, these cells that are actually encased in the middle of many layers of bone, right? You have these structures where you've got the ability to lay many layers on top of an existing structure. So you end up with this three-dimensional structure where you've got many of these cells. These cells that are in the middle are actually kind of like entombed inside of this bony matrix of stuff. And what this does is it allows the bone to grow in three dimensions. It's allowed to grow in length and also in girth. Um, we're able to grow our bone with our bodies and support them from the inside, which is not something that's really easy to do, right? And so osteichthyes is associated with this true bone structure. The teleostomy refers to the true bone, replacement bone. One way to think about this is that this type of bone is really important for supporting weight. it would be very difficult for us to get any large body size supporting our body weight from the outside, like with an exoskeleton, right? If you think about insects that have exoskeletons, they're really limited and into the size that they can actually support. The body size that they can have is limited because they're using an exoskeleton, kind of like these dermal plates, to support their body mass. Okay, we're not so constrained because we have an internal skeleton that's able to grow with us, and and that requires this three-dimensional, really super sturdy bone as we that grows as we do. Okay. The other characteristic that we find in the osteichthyes, apart from the true bone, is the presence of some type of swim bladder. Okay. Now, if you think about it, both swim bladders and true bone are going to be associated with a new kind of ecology, with a new kind of habitat, with a lower buoyant density. And so you can think of both swim bladders and true bone, this type of bone, as being associated with supporting the body weight in a new environment, a freshwater environment that had a lower buoyant density compared to the saltwater in which the earlier vertebrates had evolved. You might remember from an earlier bio class or a chemistry class that freshwater, effectively with no solutes, is going to have a density of about 1.0, effectively 1 gram per ml. Uh, Seawater is going to have something considerably higher than that because of the salts. Adding a pretty heavy load of sodium and chloride and all the other solutes that you find in seawater is going to make saltwater a lot more dense. It's going to support the body mass of these ancient fishes a lot better compared to what's going to happen if the same fish moved into fresh water. You take the same pretty heavily armored fish and move it into a fresh water with a lower buoyant density, it's going to sink like a rock. It's going to be hard for this thing to get off the ground. Um, One way to counteract that would be to put in a large sac of air, right? So if you were to say, well, I've got all these thermal plates over here. If I were to put in a big old air sac, okay, that's going to make me less dense compared to the way I was before. And so swim bladders, definitely something that we can, relate to, associate with the move into freshwater habitats. The other thing that's going to be happening is that since we're supporting our bodies with internal skeletons, being able to support all that body mass from the inside with an endoskeleton is going to require us to have some more supporting structure. So this new kind of bone, endochondral bone, is also something that you can totally associate with the move into freshwater habitats. All right, so now I'm going to move upward again. I'm going to start right here at this node in the tree of life. I'm going to be starting with the osteichthyes, and we'll be identifying a couple of more lineages here. Okay, and from the osteichthyes, we're going to be recognizing two main branches, only two. Okay, one of these is the Actinopterygii, also known as the ray-winged or ray-fin fish. Actino means ray, and pteri, kind of like the wing, like a pterodactyl. This refers to the ray-finned fish. And here we're talking about those fish like perches and like trout. I'll draw a fish for you. Okay. Friendly fish. And sure, they've got dorsal fins, and sometimes they've got anal fins. But the fins that we're really thinking about here is in terms of having rays are the pectoral fins, these fins like over here. If you look at them, they've got individual rays. If you look down here in the bottom, you've got some pelvic fins. Those also have rays. And in effect, these are kind of like the front arms, the forelimbs, and the hind limbs. These are the parts that are homologous with the forelimbs and hind limbs with the rest of the vertebrates, with the tetrapods. And osteichthyes includes the Actinopterygii, in which these fins, they've got these rays. And these rays are really kind of neat things, and they're a technological marvel because they actually create a fan-like structure which are really good for giving you a lightweight paddle that can move water really quickly without occupying a whole lot of space. Okay, so if I were going to be spending my whole life swimming, I would probably want paddles like this for my forearms. All right, so the other group of the osteichthyes is the Sarcopterygii. And sarco means fleshy, fleshy finned or lobe finned fish. Call these lobe fins. And by this I mean instead of having these ray fin, these actinopterygian fins, we've got this pretty much the same structure. We might have another kind of fish over here, and dorsal fin. Might have tail like this, right? So it might have these structures, but instead of having a ray fin, there's going to be like a fleshy lobe here, right? It's going to be it's going to have muscle on the inside. Okay, so there's muscle and bone supporting this. So basically, it's kind of like a little arm, a little paddle, a fleshy paddle. And similarly, the pelvic fins are going to be kind of like hind limbs. And a lot of these fish-like animals are going to be walking along the bottom. And so there's some animals like this. There's an animal called the coelacanth, scientific name Latimeria, named after Marjorie Latimer, who discovered this in the fish markets off the Comoro Island. She didn't really discover it, but she's the one that brought it to the attention of the Western world. Yeah, Indian Ocean fishermen had known about these things for a long time. Also, in the Sarcopterygii, as modern organisms, are a group of fishes called lungfishes. I think two species in Africa, one species in South America, and one species in Australia. So there are four species of lungfishes that still live. They've got lobe fins, and this is actually the group that's more closely related to our third group. And this is one that we care about a lot. This is what we'll be picking up our next lecture. It's the Tetrapodomorpha. And those, of course, are the ones with four tetrapod feet. These are the tetrapods. You are in this group. You are here. Okay? We're not going to get to that group too much right now. It's going to be for the next lecture. Okay. The only one last thing that I want to cover with you guys before I let you go is the timeline. And so what we're talking about here is the timeline of the Paleozoic. We did this with the plants. We said that starting from around 541 million years ago, we have the start of the Phanerozoic. Um, Before this time, we've got the Precambrian. Right? So Precambrian that way, 541 million years ago. And then we've got the Cambrian going from time A to time B. Remember, don't worry about the times. We've got the Ordovician. We've got the Silurian. Remember this from our plants lecture. Silurian is when we got the very first vascular plants. Then we've got the Devonian. We've got the Carboniferous. Thing about the Carboniferous that you might remember is that big CO2 drawdown caused by the land plants. And then at the very end of the Paleozoic, we've got the Permian, when all the continents drew together to form the great landmass of Pangaea. And then you got the break. This is the great dying that occurred here, and we've got the Triassic of the Mesozoic coming after that. So now putting together our timeline for chordates. We see the first chordates appearing for the first time in the Cambrian, first chordates. Through the Cambrian and the Ordovician, we see a whole bunch of jawless vertebrates. Now, starting in the Silurian and onwards, we see the gnathostomes, things like placoderms and chondrichthyans, as well as some osteichthyans. Now, one of the keys to understanding fully the next portion as we talk about Tetrapodomorpha and the move onto dry land is to think about what we learned during the plant lectures, how during the Devonian, late Devonian Carboniferous, we see that tremendous CO2 drawdown. Things get cooler, things get drier, right? Remember that? And so during this CO2 drawdown, we see the inland lakes, these freshwater habitats that were so well occupied by the osteichthyans. They became well adapted to life in freshwater by having this bony skeleton with true bone or endochondral bone. They had these swim bladders that allowed them to have buoyant density. They're also getting pretty good at using their swim bladders not only for supporting their body mass, but also for enhancing their uptake of oxygen. And through this period of the Devonian, we see those freshwater lakes dry up, at least during the dry season. And so we see a great need for a transition onto dry land during the Devonian. And the transition onto dry land is when the vertebrates started crawling up and becoming amphibians, at least part-time. They're part-time aquatic and part-time terrestrial, like modern-day amphibians, like frogs and salamanders. And then during the Carboniferous, I'll give you a little bit of a preview. Then during the Carboniferous, we see the very first amniotes. which are the tetrapods that become fully independent of their watery past. They basically have the amniote egg that allows them to complete their development without ever becoming fish. And then during the Permian, we see a huge diversification of two major lineages of amniotes, one of which gives rise to mammals, and the other one gives rise to pretty much everything else, including the birds, reptiles, um, three different kinds of reptiles, including the dinosaurs, of which birds are one type. Okay, so that's going to be the starting point for our next lecture.